Now, um, this is Angelina, and she's going to be talking about snake venom and antibodies. So take it away.
Thanks, Shane. Okay, so here's my paper. It's called Snake Venom Protection by a Cocktail of Verisplattum and Broadly Neutralizing Human Antibodies. You might notice two other names on the slideshow with me. That's Cecilia and Ethan. They worked with me to make this presentation for MoBio, and they had a really big hand in helping me understand this paper, so I just want to give them a little bit of credit. So what I'm going to be presenting today is a modified version of that presentation. The paper is quite long, and it covers two different antibodies that they isolated. And the methods for the second one are highly repetitive of the first. So I'm going to be mainly talking about how they perform the methods for the first. I'm going to briefly talk about the second, but if you have any questions, feel free to ask about either. Okay, so what you might have understood or might not have understood from the morning meeting video is that they found this guy, and he had a lot of pet snakes, and he kept on letting his snakes bite him because he wanted to develop his own immunity to the snakes. So scientists wanted to take a look at his blood and figure out what made him so immune to snake venom. So what they hypothesized is that over time, his body had developed a broadly neutralizing antibody against these many different kinds of snake venoms. And then for figure 1b, they use an iterative addition approach. So basically, a lapid venom, which is the family of snakes that they wanted to focus on, their venom has a couple of main components, LNXs, PLA2, and SNXs. And in order to kind of account for all these different components of the venom, they had to have a component of their drug cocktail target each part of the venom, which is what you see here. So this kind of an overview of their approach and what they thought might be the reason that this donor had so much immunity. So snaky venoming is not really the first thing you think of when you think of tropical disease, but it's actually a highly neglected field. There's 81,000 to 138,000 deaths per year caused by snaky venoming, and 300,000 to 400,000 permanent disabilities caused every year. And you might have heard of antivenom, which can kind of protect against some snake venoms, but there are a couple of disadvantages of the way that we deliver it now. First, antibodies have to be highly specific to the snake that you got bitten by. So if you got bitten by a snake and you went to the hospital, you'd have to know, you or your healthcare provider would have to know exactly which snake bit you, and that doesn't always happen. So also the way antibodies are developed now, they're caused by injecting some kind of venom from a specific snake into a different organism, like a horse. And then they would isolate the antibodies from that, but that can cause adverse reactions because obviously you're putting something non-human into your body. So that's why they kind of wanted to create a cocktail of human antibodies and small molecule inhibitors in order to effectively treat against lapid venoms. So I'm going to be using a couple of abbreviations in this presentation. So let's just talk about what these are. So NACR stands for nicotinic acetylcholine receptor, which is an ion channel involved between the connections between muscles and nerves. And 3-FTX stands for three-finger neurotoxin. I put an image of it on the board. You can see that there are these three finger-like domains, which is how it got its name. And of these three-finger neurotoxins, there are two types, long-chain neurotoxins and short-chain neurotoxins. PLA2 stands for phospholipase A2, which targets the phospholipids in the cell membrane. Lapid venom is the venom from the type or the family of snakes that the researchers wanted to focus on. And SHM stands for somatic hypermutation. So after the body essentially produces an antibody, the antibodies generate kind of random mutations over time to improve their affinity. So as the human body is continuously exposed to the intended antigen, the antibody will essentially get better and better at targeting that antigen. That's a photo of PLA2. And then I think a lot of you are from bio honors, so you probably know what an antibody is. But for those of you who don't, it's like a big Y-shaped protein. There's variable regions and control regions here, and then what it essentially does is it binds to an antigen and effectively neutralizes it. Okay, so like I said before, the researchers were trying to find the secret in this guy's blood towards neutralizing a lapid venom. So what they hypothesized is that there were broadly neutralizing antibodies in his blood that worked against many different types of venom. So here's a little bit of information about the donor. Figure C is his self-immunizations over time, and you can see that it's just giving you an overview of what different types of snakes he let himself get bit by. And then figure D is showing his immune response. So the dotted lines are controls, and then the solid lines are him. And you can see that there's a much higher immune response in this hyperimmune donor compared to any normal human person who hasn't been bitten by snakes 500 times. And then figure 1E is showing the percent hypermutated. So somatic hypermutation, as he continuously gets bit, you would expect that the amount of hypermutated antibodies in his blood would increase because they get more specific, which is what you see here, the immune versus the control. So what they wanted to focus on first were long-chain neurotoxins. And so they looked at his blood and they found all these different antibodies that were effective against long-chain neurotoxins, or they thought would be effective. And you can see that there's slight variations between some of them. Like the amino acids aren't really identical between every single aspect, but these key regions, the CDRs, the complementary determining regions, are pretty much all the same. So what they figured out from this is that all these different antibodies had come from one germline, which they called LNX-DO9. So after this, they wanted to see if LNX-DO9 was in fact effective at binding to these long-chain neurotoxins. So this is something that they performed called SPR, surface plasmon resonance monovalent affinity, which is similar to another method that we're going to talk about later called biolayer interferometry. But the idea is that you fix something to a sensor, in this case the antibody, and if the antigen binds, it'll measure like a shift. And this steep line you see here is binding, and then the plateau is steady state binding. So this is really good because you want your antibody to be able to bind securely to your antigen, and then you can see that DO9 does just that. It doesn't have like a drop or anything, which would indicate dissociation. So they wanted to keep on testing if it would bind securely, and here they perform biolayer interferometry. The two different lines are different concentrations of venom. Red is 500 and orange is 250 nanomolars. And then black is the negative control. They found, you can see there's a pretty significant difference here, indicating that the antibody did bind pretty securely to the antigen and stayed there, which is the important part. So in order to make sure that these results weren't a fluke, they also tested the antibody against venoms that didn't contain LNXs. So theoretically, if a venom doesn't contain LNX, you shouldn't see this kind of clear binding because there's nothing to bind to. And that's what they found. You can see for all these snakes that don't have LNXs in their venom or don't have a high percentage of LNXs in their venom, you don't see as much binding compared to the graphs that look like this. They want to confirm the same thing again, so they prefer immunoprecipitation. The idea is that the antibody binds the antigen and then everything else is pretty much washed off. And then if the intended antigen stays there, that's a good indication that the antibody is properly binding. So you can see for these snakes, the LNX remains, and then for these snakes, it doesn't. And those are snakes that don't contain LNX in their venom, so you obviously wouldn't expect an LNX to be there. So these little spots in the gel, which they thought were LNXs, they sequenced and confirmed to make sure that they actually work. So now that researchers had pretty strong evidence that DO9 did actually work by binding to these long-chain neurotoxins, they want to take a look at exactly how that binding was happening. So they created a couple of crystal structures, and this, in my opinion, is the most interesting part of the paper. So A, you can see the interaction between DO9 and a lot of different types of toxins, and you can see that toxins look pretty different, right? But they all bind to the same region of the DO9, which is the most important part. And you can see in B and C, here's a kind of zoomed in perspective. You can see DO9 binding to cratoxin and NAC-R binding to cratoxin. So I was supposed to mention this before, but the way three-finger neurotoxins work is by effectively acting as a competitive inhibitor and bind to NAC-R. So essentially you don't want these venoms to bind to your receptors. And you can see here the interaction between DO9 and the toxin and NAC-R and the toxin is highly similar. It binds to pretty much the same region. And you can see it in this comparison as well, figure 3D. The area where DO9 and NAC-R bind to the cratoxin is really similar, and they look really similar. So that's not a coincidence. The thing about these toxins is that, yes, over time they can change and become different versions of themselves. But the fatal flaw of these toxins is that they need to be able to bind to NAC-R. So because NAC-R doesn't really change, they have to have one conserved region on the toxin that is able to effectively bind to NAC-R, otherwise they don't function. So you can see here in figure G The white stands for 100% conserved, and then the purple is 0% conserved. And you can see these regions where NAC-R and DO9 interact with the toxin are the most conserved regions. And this is really significant because this allows the antibody to bind to a region of the toxin that will essentially always be there. So by targeting the same region that NAC-R and the toxin interact, the antibody is able to be effective across many different types of toxins and over time still be effective against toxins. And then figure H is just showing you which amino acids it's interacting with, and you can see that for DO9 and NAC-R, they interact with really similar amino acids. So all this information is really great. We know that DO9 binds to the long-chain neurotoxins, and we also know that the crystal structure, based on the crystal structure, DO9 and NAC-R interact with pretty much the same regions. But this information isn't really helpful if it doesn't actually improve survival rates. So that's what they decided to measure here. In figures A through G, they first mixed the toxin and the antibody and then injected it into the mice. And you can see for the majority, like B, C, there's 100% survival rate. So it's really effective at keeping these mice alive. But that's not really the way you get bitten in real life. In real life, if you got bitten by a snake, you'd get bitten, you get sent to the hospital, and then you get injected with something. So that's what they decided to test. They first injected the mice with snake venoms, and then they waited 10 minutes, and then they injected them with the antibody. And you can see here that the solid lines are the injected with the antibody group, and then the dotted lines are the control group. And the antibody was really effective at causing survival. You can see it's all 100% survival. So here's what I was talking about. They also performed all the same procedures, and they found this other antibody called SNXBO3, which is effective against the short-chain neurotoxins that I talked about earlier. I'm not going to go too in-depth about that because you can see it's all kind of looks the same. But something to really note here is that, remember how for the SPR graph of DO9, it was really flat here? You don't see that with the BO3. Here, there's a drop or a disassociation, and you can see for the KD constants also, the graphs where the LNXs were all like in the pico, which is smaller, and then you want your KD to be smaller because that indicates higher binding affinity. So BO3 isn't really as effective of an antibody as DO9 is, essentially. And you can see that in some of these bilayer interferometry graphs too. Like for this one, it's supposed to be effective, but you don't really see that much of a difference. Okay, so after that, they wanted to test what they were originally trying to create, which is essentially a cocktail, a mixture of DO9, BO3, and a small molecule inhibitor called verisplatib, which is effective against the phospholipase A2. So they tested a bunch of different mixtures of this. They tested just DO9, just verisplatib, DO9 and verisplatib, and then the combination of all three. And then black glide is the combination of all three. So you can see for some snakes, it was really effective at prolonging the survival of mice. C, E, F, G, they all caused 100% survival. But for other snakes, it's a little bit weird. For B, H, I, these mice are dying, and that's not good. And the reason that they identified for this inability to effectively protect against some types of snake venom is the inefficacy of BO3, because it doesn't bind as well, and it's not really as effective. But overall, for the overwhelming majority of snakes, this drug cocktail was really effective. You can see for the cocktail with a mixture of all three, it provided 100% protection against all these other snakes, and the ones that didn't still provided 40 or 20% protection. So this is really significant because it doesn't require you to know exactly which snake bit you. You can kind of use this universal human-derived cocktail to treat yourself, and it also works for snakes of many different parts of the world. So it's not like regional or specific. I just want to thank the members of Journal Club for helping me make this, the leaders, and also my team, Valentina, Catherine, Shay. Valentina, Catherine, Shay, and thank you for listening.
guys could scan this for your attendance, that would be great.
We don't have time for questions? Student questions?
Yeah, if you
Oh.
want to answer questions.
Yeah, I'll answer questions.
Yeah, for sure.
Does anyone have any questions? No, we're just in attendance.
Yeah.
I mean, there should be 8 questions.
No students have questions?
Oh, also say there's a bunch of questions.
Oh, there's also food in the back.
We should take a question from, okay, there we go, Joe.
So how did the guy who got himself bitten by snakes, how was he not dying like when he first got bitten by a snake?
He did. He got sent to the emergency room. He went into like a coma, but yeah, over time he just got better and better. Oh,
So, like last slide you showed, like that showed like the survival rate for like all the different types of the cocktail. Were those also tested after 10 minutes, or were those like the immediate?
I believe those were the immediate.
So they didn't test them after 10 minutes.
Oh.
Test for like hours after, like sometimes if I need to get to the hospital.
They tested for a couple hours. It goes up to 1,500 minutes. So it's like a little over. I can't do math in my head, but they tested it for a while, but they didn't really test it after that. So, but for some of these that had 100% survival, it maybe isn't as important.
Do you think that they included the VO3 in the mixture so they found that it wasn't as effective?
I think they included VO3 because if they didn't include it, that would have been worse. Like it's better to have like an antibody that has some or partial protection rather than just ignoring that part of the venom, because it's a pretty substantial part of the composition of a lapid venom.
So what do you think the next steps would be for this research in terms of like, do you think, and or have you looked into anything that they are currently working on for this, maybe with respect to VO3?
Oh, I have not looked at that, but in the conclusion, they mentioned that they want to kind of test it on bigger animals because mice and humans, maybe there's not as big of a correlation. And also you're injecting mice with human antibodies, so maybe it can be more effective in people. And then also, I feel like this iterative addition approach could also be like applied to other different types of venom as well, not just the lapid venom. Okay, thanks everyone.
Do we have a next?
Oh, please put